Uh, can you please let me know whether whether you can hear me? Okay, good. So,
Recording in progress.
we can start then. I hope you can hear me. Right. So I'm going to take the third lecture in this biology one. Uh, okay, we were talking about the cell structures. So we have seen the animal cell and also the plant cell. I said the difference, the main difference is that plant cell has a cell wall and a vacuole and chloroplasts. And then subcellular structures we studied. So several of those we discussed last time, and their functions as well. So I was talking about ribosomes. Now ribosomes, they are the cell's protein factories, because the cell produce the protein synthesis is the function of the ribosome. The protein, when you take, we synthesize our own protein. We eat protein and we digest it, break it down and create amino acids, and we absorb amino acids. There are 20 different amino acids and we put them in the order according to our own DNA. So this is the protein synthesis. I will explain you later in the section. But just for the time being, remember that ribosomes are the centers for protein synthesis. So there are three ribosomes, just three in the cytoplasm, and they produce proteins, and these proteins are used for cellular activity. But there are ribosomes which are attached endoplasmic reticulum, or called ER, and these are called attached ribosomes. And when you take ER or endoplasmic reticula, because of the attachment of ribosomes, there are two types of names for so endoplasmic reticula are they are like the road system inside the cell. Things can be transported easily through these endoplasmic reticula. So if the reticula is singular, reticulum is singular. And if the ribosomes are actually they are all up in the plasmic reticulum. And if they are not attached, they are called smooth endoplasmic reticulum, right? So the smooth endoplasmic reticulum, CR, there are some more functions like lipid synthesis, and also they assist in detoxification to break down toxic compounds, and also they act as calcium storage. So then nextly, we were talking about how the proteins are going through endoplasmic reticulum. So you can see here, ribosomes are attached. These ribosomes function means synthesis of protein. When the proteins are synthesized, these ribosomes release those proteins to the cavity or the lumen of endoplasmic reticulum. So you look like this: one ribosome there, and this ribosome produce protein, and these proteins are released to the inside of endoplasmic reticulum, inside this cavity or a lumen. So these proteins are transported through the endoplasmic reticulum to the end of the reticulum. And at the end, and create these vesicles. Then these vesicles bud out. Vesicles bunches from the ER or endoplasmic reticulum, and these vesicles you can see now. These are small bubbles inside the cell plasma, right? Cytoplasm. and these bubbles have inside you get protein, and these are passed to these are sent to the Golgi body. So I showed you then again the Golgi body. Vesicles are coming from endoplasmic reticulum with protein, and these ones come and join the endothelial Golgi. These join that give that protein to the Golgi apparatus. Usually, Golgi apparatus is situated near the cytoplasm. When we want to export protein, you send it to the Golgi body. So these proteins are the various things are added to these proteins inside the Golgi apparatus. For example, Golgi apparatus can add different minerals or carbohydrates or lipids or other things to these proteins, and then they just export it from the cell. Or they again make vesicles from the ends, and these new vesicles you can see these vesicles shipping inside of Golgi apparatus, shipping inside whom they are sending on. So they are sending and go and attach to the cytoplasma membrane or cell membrane and put these things outside. These outside, these proteins are collected. For example, if you think now all the enzymes are protein. So when we eat something, also the saliva, we secrete saliva. They also you get inside. Secrete saliva means saliva coming to the mouth. So this saliva, proteins in this saliva are produced by ribosome. Ribosome send those proteins to the Golgi bodies through endoplasmic reticulum, and Golgi bodies uh, send it outside the cell. For example, in the mouth, you get either side of the mouth, you get parotid glands. In parotid glands, those cells, this happens. So they are Golgi body, send these whatever the saliva or the highways and send outside the cell. So you have just meant that the function of the Golgi body, or the Golgi apparatus, is secretion. Also, it has another concept, right? Also, that it says also is not clear. So, you know, I would say earlier is not clear. Is it okay now? Hello? Clear to you now? it clear now? Can you hear me? Can you hear me now? Okay, thank you very much.
Recording in progress.
So, proteins produced in ribosomes go through the endoplasmic reticula. At the end, they form bubbles. So these bubbles are called vesicles. They are coming with the protein. They are sent to the Golgi apparatus, and at the end of the Golgi apparatus, again the bubbles are formed and they are sending outside. Within the Golgi apparatus, lot of things are added to those proteins so that the protein uh, they can make in whatever the manner they like. So I was telling you about the parotid glands in our mouth. You see, either side of the mouth you get parotid glands. They are the secrete saliva. The saliva has amylase. It's a protein. So those are protein. Those proteins are secreted by salivary glands, glandular cells in the parotid gland. So what they do is they produce protein and into the body apparatus and then send to the outside. That protein is amylase. It is the saliva. So likewise, cells have to produce proteins and send outside. So they are all our digestive enzymes. So likewise, send outside the proteins they produce. So then this again shows the same thing. Ribosomes produce proteins and through GR, they send it to the vesicles, they send to the Golgi body, send to Golgi bodies to and put it outside the cells. This is called exocytosis, going outside the cell. And then this one last slide. So that lysosomes contain these are digestive. Can you hear me now clearly? Can you hear me? Can somebody? Hello? Yes. can see the you can see here endoplasmic although they look separated, they are actually joined. These all because in the cross section they are not joined. Means this cutting plane they are not joined, but they are joined. I told you to take 4 fingers like it looks like these four balls here. There it looks like they are not joined. Four balls, but they are connected from here. One connection. All the correct. They are joined. So just I now I will continue. If it is not clear, please let me know. So this is again these produce lysosome. Lysosomes are they have digestive enzymes. So inside the cell, they can if these small vesicles, they are also enzymes means they are protein or they are protein inside protein. Fiber produce the protein. These produce are sent to the world. Both bodies make these lysosomes. So these are the lysosomes you can see here, and these lysosomes they can to digest things. So these lysosomes have few enzymes. So if they burst, what will happen? If they burst, if the lysosomes burst, cell will digest because it has got of digestive enzymes. Lysosomes are like little bombs. They are like bombs, right? Inside the cell. If they burst, or the whole cell will die. So they are again, they are also surrounded by special by layers, special type, so that there is no leak. If they leak digestive enzymes, then it will digest, the cell will digest. So what they food particles. Here I have shown they are taking in food particles. They are taking in this is endocyte. Endo is taking inside. You can phagocytosis is also taking inside. When they take in the food particles, they make a nice vacuole, bubble like this, so that this is the food vacuole. These vacuoles are again coming from the wall of the vacuole. The wall of the vacuole made up of the cell membrane. Thus, this originated from the cell membrane. So this has the cell membrane surrounding it. So these vacuoles come and lysosomes go and attach to them. Then they are attached to the food vacuole. They release the digestive enzymes to the food vacuole. Then what happens? Food is digested. They are digested. This juice is absorbed. Digested cell can absorb and cell can remove exercise by exercises the things which are not digested, the things which should be digested, those things, the remaining stuff they remove. This is how cells can digest. Cells can eat or take food inside and digest lysosome, right? Okay. Next is this one. I have this one diagram I have given you. Again, the same thing I'm just trying to say because I'm telling you again and again. This again, ribosomes uh, protein. These proteins come to the vesicles to the Golgi bodies. Now, Golgi bodies they produce lysosome. So just now, when they take in foods like food vacuoles, they can join the lysosome to food vacuole and digest the food, right? This is very serious because the lysosome, if they burst inside the cell, if there is a leaking, these enzymes can digest the cell. They make sure that the enzymes are not leaked outside. They just come and put the enzymes only to the inside of the food bacteria. So the food is digested and it can be absorbed. Also, autophagy, auto means by it itself mean signal what we call the direct transmission is via. That means તો ક્રમાંકનો એના ભાગને કરવાનું બોલવાનું છે એને કઈ autophagy means. Autophagy. It can digest cell organelles which are no longer needed and absorbed to cytoplasm. Now, suppose there is a mitochondrion. It is very old. It is not functioning. It is very old. The cell does not need this mitochondrium anymore. So what happens? They send a lysosome to the mitochondria and digest it and absorb it. Right? Likewise, they can do this same digestion for the cellular structures also, which they also, this ribosome can burst and digest the whole cell. This is an example I tell you. You know this frog. Frog, it's like the larval form of the frog. We call tadpole. That is a calling signal. Scadia. Scadia has a tail. And when it comes to the land and become a frog, the tail, they digest the tail and absorb. They are all the lysosome in those cells. So all the cells are digested then. The tail is no longer there when the tadpole become from. So this is another example for lysosome. Okay. Next we are going to another organelle that is called centrosome. Centrosome. Some means body, right? Some means body. Centro means center. Now you can see in the cell, centrosome is located in the central region near the nucleus. Then we draw diagram, cell diagram. We draw a cell, centrosome near the nucleus in a cytoplasm in the middle of the cell, sort of in a central site, right? Central area. So the centrosomes, if you look at, they are made up of centrosomes are made up of two centrioles. There's only one centrosome usually. They can duplicate during the cell division. I will tell you that later. In the centrosome, only one, and actually to take that one, it is made up of two sub components. They are called centrioles. These two centrioles are situated at right angle to each other. You see, these two are at right angle to each other. So if I go just a small description about the centrophore I have here, to find these centrosomes, still depending on the cell, and they are not surrounded by a membrane. And with two centrioles, which are made up of microtubules, centrosome is made, there are two centrioles. These centrioles are made up of you can see structures that in the exome, but are called microtubules, right? So these are made up of microtubules. This involves in spindle formation during cell division. Before cell division, centrioles duplicate. Get it? Next, we will do the cell division. When I do the cell division, I will explain this little bit more. Okay, so this is centriole, another type of cell organelle situated in the central area of the cell. Now this is a cell division, cell division here. So just before cell division, they duplicate. They duplicate means centriole, centrosome divide and make two centrosomes. These centrosomes go to two ends. These are called poles. Pole means dravaya, right? Drava Dakshina Drava. Likewise, when the cell is dividing, they first mark the two poles, North Pole and South Pole like that. These are not called North and South. They make two poles like this. And this is the centriole. It makes these spindles. You can see these fibers like structures. They are spindles. These spindles attach to chromosomes and take them towards these poles. I will explain this later under cell division. Right? Now then again, in the plant cells, we have the cell wall. Now, as I told you earlier, also I told you that cell wall provides the shape to the cell of a plant cell, right? And protection and support. So the animal cells, they don't have a proper shape actually. They have a shape, but that is not very rigid shape. Rigid, not rigid means you can change it. If they need, they can change the shape. But plant cell, they have a definite shape. So because of the cell wall, and also cell wall gives the protection. The cell wall is made up of carbohydrates and protein fibers. So mainly this carbohydrate is called cellulose. We all know cellulose. So the cellulose is the major carbohydrate component found in cell walls. And then the other structure here given in like a small swimming pool inside the cell, this is called vacuole. In this vacuole contains liquid. This is called plant sap. Uh, and it acts as a food storage and also as a waste material storage. So it can store food material and also it can store waste material until they are broken down, right? So those two things are the functions of vacuole. So cell wall and vacuole. Then you can find cilia and flagella. Cilia are the things you can see in the this is in fact this cell has lot of these fibers like things coming out. They are cilia or flagella, right? If they are long, very long, you call it a flagella. They are small, then we call cilia. They are also made up of microtubules. Microtubules, you remember, you saw in the centriole or centrioles, and can be used for locomotion of a cell. Locomotion means what? Motion, motion means, motion. Locomotion means animals can do locomotion. Locomotion means going from one place to another place. Now, plants can't locomote. There's no locomotion for plants. Plants can't go from one place to another. But animals can go. We can go from one place to another. We can walk. Like we have legs. Now these single cellular animals and usually they have structures. Using these, they can move. They can move. It's like a boat. So with the habala you can move, right? So these act as they just by waving these cilia, these cells can move. So can be used for locomotion of a cell means that going around using these, right? Can be used to propel a layer of liquid outside the cell. So to take this example here, what I have shown you. So you can see the cell layer with the cilia. Now, this is a type of cell layer, epithelial, the cell layer we find in our respiratory tract. In respiratory tract, you see, this is the mucus. Sometimes you call this from the respiratory tract, you get this mucus back to your mouth. We call it කියන්නේ අපි සෙම එනවා කියලා කටට සෙම කටට එන්නේ ගාපුවාම කාරණකොට මෙහෙම උඩට එනවා සෙම. Now that purpose also this respiratory tract, this our respiratory tract has the epithelium or the outer cell layer as cilia. These cilia can push this slime layer or the mucus right towards the mouth. Now this is the layer and they push it and go beyond and again push it. It's like when you swing, you put the effective stroke front and then take it with the hand without giving a effective stroke. You understand what I say? Now when you put even the first stroke when you push the water and then when you take the hand back to the original position, you are not you trying to make the movement of the water. Right? The effective stroke is the one you push, right? Twice here also push it and take it back slowly and then again push it, right? So likewise it can push the propeller layer of liquid outside the cell. This is the function of celiac flagella and this is the electron microscope photograph of a lung. Bronchial wall is the what I tell the respiratory tract, right? We call bronchus and bronchial layer. They are the surface if you take in the cavity. If you see the surface, you can see a lot of cilia coming out from the cell. These cilia will move and send the mucus towards the mouth in certain circumstances. And also flagella. Here I have shown the picture of sperms. Now sperms have flagella. These sperms are used to swim for sperms, so that it is a locomotion of the sperms. Locomotion means swimming from one place to another place. Okay, so cilia and flagella are there for movements. So that the function of that is the movements. Then the nucleus. Nucleus is surrounded by a nuclear envelope. So you can see here, I will explain this nucleus here. This is the nucleus. Plural is nuclei, L-E-I. So these are Latin words, Greek words, Latin and Greek. Not like English. In English, nucleus should be the plural, should be nucleuses. But here, because Greek word, we call it nuclei. That is how the plural name. N-U-C-L-E-I is the plural. Okay, nucleus. So you can see the nucleus. This is inside the cell, in the middle of the cell usually. You can see there is a nucleolus. The function of the nucleolus I have given here. Nucleolus produce ribosomes. So this nucleolus function is production of ribosomes. And the nucleus is surrounded by the nuclear envelope. This you can say nuclear envelope. Nuclear envelope is made up of two layers of cell membrane. Again, that our famous phospholipid layers, cell membrane, two layers of phospholipids. I said. And nucleus surrounding the nuclear envelope has two layers of cell membrane. And then from places you get in some places pores. Pore mean yes, these pores like things, right? So that things can move in and out through these nuclear pores. And you find chromatin. So that word chromatin, okay, we'll go one by one. Here in the description, surrounded by a nuclear envelope. Nuclear pores facilitate movement of molecules in and out. Nucleolus produce ribosomes. Chromatin is made up of DNA and proteins. During cell division, chromatin become condensed and chromosomes become visible. So during this is all, and also you can see endoplasmic reticula are sometimes attached to the nuclear membrane like this, and those endoplasmic reticula you find these what I have shown here are rough endoplasmic reticula because ribosomes are attached. Now these chromatin I'm talking about, here chromatin, right? That is chromatin is you get lot of things like you get chromosomes here, but they are at a very dispersed state. They are diluted like dispersed. But during cell division, they condense. They condense and make molecular structures called chromosomes. We will go and see the chromosomes next. So inside the cell, during the cell division, you can see very clear structures like this. These are chromatin. Chromatin, now condensed chromatin, right? Condensed chromatin, concentrated condensed chromatin. These, otherwise, when it is not dividing, when the cell is not dividing, these chromatins are not condensed. But when the during the cell division, these are condensed, and we can very clearly see now chromosomes. Now, when you take a chromosome, so this is the nucleus. I have just taken a picture. You can see two layer. The envelope has two membranes, and this is the nucleolus which produce ribosomes, and the nucleopores are there, right? So if you take a chromosome is one structure like this. This is a single chromosome, and in the middle you can see a sort of swelling, and this is called centromere. Centromere, the central, you can see centromere. So this is a chromosome, and you can see the centromere. This chromosome has DNA, and this DNA duplicated into two chromosomes. So this is how it happens. When it two chromosomes they are making, you can see this divide into two and make divide means it is not dividing, it is duplication. So because of the duplication, now you can see two chromosomes and still they are attached from this place. It is centromere. So these daughter chromosomes called, these are sister chromatids. These are called chromatid. This is a duplicated chromosome, and when they are duplicated, they make two structures like structure. Like the mother, they make two daughters, right? And these are called sister chromatids. You can see they are still attached to the centromere. So the chromosome structure in the diagrams, you can either go like this or like this. Most of the places, chromosomes are drawn like this. It's just to show that chromosomes are duplicated very often. That is why this is the comes. So either way you can draw it. Okay, we will go to see this chromosome. So cell, inside the cell you can get the nucleus, and inside the nucleus you can get chromosomes. These chromosomes cannot be seen during the normal time, but during the cell division, very these are important there. You take a chromosome, you take inside the chromosome, you take a long thread like long thread. Thread the DNA molecule. But this DNA molecule, this is the DNA molecule. This DNA molecule, I will explain you later. You see, this is the DNA molecule. This DNA molecule is wrapping around or woven around protein balls. These protein balls are called histones. So histones are protein balls. So these protein balls, this DNA molecule is wrapping around protein balls and make this ball-like structure, right? These ball-like structures are called nucleosomes. Some again a body, I just said, right? They here we call it some in they here. So nucleosome is fine. So these are nucleosomes, right? These because some word come always bulbosome, ribosome, centrosome, nucleosome. So just means body collective, right? So you can see DNA molecule, long DNA molecule, one molecule here, only one molecule. This molecule has a long thread, but to pack it nicely, they have is woven around 14 bowls, and there are 8 protein bowls in each big bowl here. In each big bowl, you find 8 protein balls. So these big bowls are called, histone protein. Sorry, histone, 8 histone protein molecules are there, and they are woven around, and these balls are called nucleosomes. Again, these balls are also very tightly they bind or the woven around, and then ultimately this make central cell chromatic or the chromosome is made up of like that. So chromosome is made up of like that. So you can see inside the chromosome very long DNA molecule is there. Since the DNA molecule is very long, you have to pack it properly. To pack it properly, this DNA molecule is woven around protein balls called histone protein molecule, and these they make balls called nucleosomes. These nucleosomes are again packed very tightly to make the centrosome. So this is the sorry, not centrosome, chromosome, right? So this is the structure of the chromosome. Inside the chromosome, you get the basically long molecule of DNA molecule is double stranded. I will come to DNA later and explain after molecular biology. So I'm not going to touch this structure now. Okay, we will go to again these chromosomes. Chromosomes, you see here in the cell, how many are here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. This has 12. In our human cell, how many chromosomes are there? There are 46 chromosomes in the human cell. These are the now the scientists have mapped and they know all these chromosome structure from top to bottom, right? They know very well. So this is a diagram of those chromosomes. Some are short, some are long. In some the centromere is in the middle. In some the centromere is towards the end, right? So these are the structures of our chromosome. So they are in pairs. You can see they are in pairs. So when you take, we have 23 pairs. We have 23 pairs of chromosomes in our cell. In each cell, in one cell, you find 23 pairs of chromosomes. So one pair is different from other pairs, and these pairs determine your sex. This pair determines your sex. Right? This pair, the members are called, if it is female, these are XX. X chromosome, this is X chromosome. So two X chromosomes are there in a pair if you are a female. If you are a male, you get one X chromosome and another different one, short one, and it is called Y chromosome. So in females you find XX and in male you find XY. So the 23rd pair XX or XY. All the others, the both are the two members in the pair are similar, right? These are called autosomes. This pair is called sex chromosome. Right? This pair is called sex chromosome. It can be XX in female or XY in male. All the other 22 pairs are called autosomes. Autosomes mean they are not involved in sex determination. They are called autosomes, right? So you have 23 pairs. Now pairs, why pairs? Because the same chromosome, number one chromosome, the same chromosome, you have got one from mother, one from the father. So you have got two from each type. That is why the two from each type. Ninth chromosome if you take, one has come from your mother, and the other one has come from your father, right? So that is why you get. So we have basically 23 types and 46 altogether because from each type you have two. One has come from mother and one has come from father. Okay, we go and see the Freud level. Freud means the Freud mean like chromosome or the thread or the structure, whatever the Freud is that you use it for a sort of membrane thing, but here this refers to the each is called one Freud, the Freud structure. One structure is Freud, okay? Diploid means you get two types. That means that what I showed you in the cell, human cell, 23 pairs. You see, humans, 2M. There are 23 types, but two from each type. So altogether 46. 23 pairs. So diploid means two copies of each chromosome type present. One from mother and one from father. Members in a pair are called homologous chromosomes. These members are called homologous chromosomes. If you go back, these two are called homologous chromosomes. These two are homologous. You can say this one is homologous to this one. This one is homologous to this one in each pair, right? Okay, two copies of each chromosome type present. One copy is coming from mother and the other one from father. Members in the pair are called homologous chromosomes. So humans have 23 homologous pairs. That means altogether 46. We call it simply 2N. 2N is the diploid. Diploid means two members from each type. And you can get haploid. Haploid means one member from each type, one from each chromosome type. So since it is one, we call N. And polyploid, you can get more than two also from one type. For example, if you take wheat, the plant wheat, it shows hexaploid. Hexaploid is one. Six chromosomes for each type. So you call it 6N. Actually, they have how many types? They have 21 types of chromosomes. But from each type, they have six numbers, so that altogether they have 126. These are not very common. These are common only in plants, not in animals. Right? Animals, you always get 2N, and N also you can get in gametes. I will come to that. Okay. So this is the priority level. Now we are coming to cell division. Cell division, there are two types, mitosis and meiosis. Mitosis and meiosis. We take one type and see first, right? This is mitosis. In mitosis, what happens? In mitosis, what happens? Mitosis, during the cell division, I said, before cell division, this uh, centrosome duplicates. Centrosome, remember, it duplicates, and each cell duplicates then moves to poles, moves to the poles to mark the pole. Right? I said Uttaradhrave, Dakshinadhrave, likewise, they go to either end to make the ends, right? So that it has gone to the ends. And in this cell, where we take this example, you have four chromosomes, right? Four chromosomes, and during the cell division, they all come in a line in the middle, and this forms these fibers like spindle. They're called spindle, right? This spindle are made up of microtubules. These microtubules bind to this one. Now this one is a chromosome. This chromosome is made up of two chromatids. These bind each other and separate these two chromatids, right? Separate these two chromatids, and they are separated now, and pull them to the two ends, pull them to the two poles, right? So that these spinals get shorter and shorter. When they get shorter and shorter, these are pulled. These are pulled from the middle. Because of that, you can see a V shape in the chromosome in this stage, right? So at the end, these are pulled down to two ends. You can see here now, they at the end they form the nuclear membrane around that. You can see two nuclei appear. Nuclei is the plural, right? Nuclei appear. You can see now each uh, nucleus you find four chromosomes, and cleavage furrow goes here, so the cell divides into two cells. Okay, we will take different stages for names. Don't bother too much. I'm just very simply giving. This is a prophase. Phase is the stage, so prophase is the before, the one before. Metaphase, and anaphase is towards the end, and telophase, telo mean in usually in Greek, antaya, right, in Latin. So antaya means towards the end, right, towards the end, telophase, right. So that is the end of it, right. It is mitosis, very simple. The cell, I will tell you again, the cell had four chromosomes. These first the centrosome divide into two and go to mark two poles, and four chromosomes line up in the middle, and these microtubules join to these chromosomes, and they separated chromatids from each chromosome and pull the chromatids towards the poles, so that at the end they surrounded these chromosomes again with the nuclear membrane and two nuclei. Now this cell is can divide. Cytoplasm is now dividing into two, right? So two cells are formed. This is how the cells are dividing. This cell division is very simple, and this is called mitosis. You understand that, I guess, right? Then we go to, there's another type. Mitosis, this happened. We had a diploid cell at the start. It had 46 chromosomes. And then in humans, in our cells, we have 46. When this cell divides into two, this one also get 46. This one, each daughter cell get 46. This one divided into two, and each get 46, 46. Right? So there are instances. So for example, I'll show you this. What is this? A rocket going to the moon. Huh? This is a sperm coming to the ovum. This happens, fertilization. When the father's sperms and mother's ovum, they join together. If this has, what happens here, sperm plus ovum make a zygote, one cell. That one cell is called, in Sinhala we call Yuktanu. Now, it is in English we call zygote. Sperm and ovum come together, join and make the one cell zygote. So what will happen? 2n plus 2n, so that will give 4n. This never happens in animals. This never happens. 2n and 2n, 4n. Polyploidy you can't see in animals. Polyploidy, under general normal conditions, you can't see in animals polyploidy. So there should be a mechanism to half the number of chromosomes during just before gametes formation. These are called gametes, Janman. Gamete, sperm is a gamete, ovum is a gamete. So when the gametes are produced, there should be a mechanism to halve it. So if N comes from sperm, if another N comes from ovum, the zygote can get 2N. So this half, how the gamete formation, this halving process is called meiosis. This division is called during gamete formation. This division is called meiosis. Now during meiosis, diploid 46 divide into two, and there is a there are two cell division in the meiosis, right? Meiosis two cell division. At the end of two cell divisions, always two, and at the end you get four cells. These four cells have half, that is 23, 23, 23, 20, right? So that these gametes, sperm or ovum, when they join to again, two then come together, you can get 2N. N, 2N you can get, because this is N now, haploid. Haploid means one chromosome from each type, 23 types. When these two gametes join together, then you can get diploid 46. So we will see meiosis, the process of meiosis. This is meiosis. In this meiosis, what happens is, okay, hello, can you see the screen? Can you hear my voice? Hello? Can somebody answer me? Can you, is something went wrong here? That's why I'm asking. Okay, I will continue this, right? Okay, you can hear, good. Right. So just let me continue this a little bit more. This is meiosis. That is half. How you may going to half this? Okay, this is the way you make the half. This division is called meiosis. Now in meiosis, this division, you see again the centrosome is divided into two and make two poles like Uttaragravya and Dakshinagravya, whatever. Don't use these words Uttara Dakshina, but these are poles, right? So they here in these chromosomes can come as a pair. As a pair, they come to the middle and make a line. So when they come to the middle as a pair, in the first division, it takes the whole one chromosome to one side, the other one chromosome to the other side. So for example, there are two pairs here. In this pair, one may be this lighter one may be from mother and darker one from father. In here, the darker one from father and lighter one from mother. And then when they take towards each side, now they are not taking chromatids, they are taking chromosomes. One pair they are separated. When one pair is separated, you see only two chromosomes are going to each end. To each end, two chromosomes are going. And then they go for the next division. In the next division, two chromatids are separated. In next division is similar to mitosis. This is similar to mitosis, where in each of these two chromatids are separated, right? So at the end, you get four cells. Each cell has only two chromosomes, not four chromosomes. The mother cell or the parent cell had four chromosomes. Here now you get only two. One chromosome from each pair. You understand? This cell division is called meiosis. And the earlier one was called mitosis. In fact, the meiosis has two cell divisions. In the first cell division, chromosome pairs are the members of the pairs are separated. In the second division is similar to mitosis. Similar to mitosis, from each chromosome, chromatids are separated. So you end up with four cells like this. So this is meiosis. So I told you mitosis and meiosis here. Meiosis happens only during the gamete formation. So these are four cells here are gametes. They are gametes, either sperms or ovum. Sperm or ovum. Now, when they join, one sperm and one ovum join, they can again make two M. Two M they can make because another member come for the from the same type, so that they can two pairs like this, right? Two M. These are haploid. This is N. Then there comes two N. So this is meiosis. So you here learn a big story here. Cell division, two types, mitosis and meiosis. And this is the mitosis I explained. And these two cells join. Because of that, we have to half it so that we have another type of cell division for gamete formation that is called meiosis. Right? This is meiosis. So the key differences between mitosis and meiosis are given here. Mitosis, what happens, occurs in all somatic cells. Okay. Somatic cells mean sperms and ova, they are called gametes. Gametes they are called, right? All the other cells in the body are called somatic cells. Somatic cells are all our body cells, right? Occurs in normal cells. Mother cell is diploid, 2N, okay? Two cells are resulted, they are also 2N. One round of division completes the cycle. And in meiosis, what happens? Occurs only in gamete formation. Mother cell is diploid, four daughter cells are haploid. Now in two rounds of cell division complete the cycle. Actually, second one is similar to mitosis. Okay, so you have come to the end of the cell division. Okay. Crossing over. There is a process called crossing over. If I going to teach you that, that is going to be a bit complicated. Then you don't have to worry about that. So just remember that during meiosis, crossing over means exchange of parts between different chromosome pairs. This can happen because pairs come together. If you remember meiosis, pairs come together in the middle during the first the lineup. They line up in the central line. So during when they line up, one pair parts can go one chromosome in one pair. Some parts can be exchanged. This is called crossing over. Puanti madam will explain little bit about crossing over, I guess. And just remember that crossing over can happen during, or the exchange of chromosome parts can happen during meiosis. So this is the cell cycle. Cell cycle, you can see this is the cell division. When the cell chromosomes are separated and divided, you get this thing. Again, the DNA synthesize. This one chromosome make chromatids like this. And then at the end, chromatids are separated. This is a mitosis I'm just showing. Chromatids are separated. It is a cycle is going there. This make two cells again enter these new cells enter to this cycle and go and separate, and again this one go. So cells divide, cells keep dividing. So this is called cell cycle. If you look at this cell cycle little bit more detail, you can see the chromosome separate, single, and synthesize or duplicate and make two chromatids. Two chromatids come together, they are separated, again make single, likewise it is called cell divides, right? And this is cell cycle, little bit detailed manner. Here you get interphase and mitosis. The mitosis is here. Mitosis, this is the division period. Division period, I said, don't worry too much, but I told you these basic words. Four phases are there. Prophase, metaphase, anaphase, and telophase. These four phases are there. And then new cell goes to a GO is the period where the normal cell doing its normal functions. So they have some functions, so they do functions. And then they come to gap 1, preparing for DNA synthesis. and then the replication occur, DNA replication. This is called synthesis period S. And after replication, now they have two chromatids, right? Two chromatids, one chromosome, now two chromatids, each chromosome. And preparation for mitosis, and the growth also happens during this period. This period also growth, and they enter the this phase. Then cell division occurs. Again, the new cell comes, go like this, and at the end, that cell also go to the division period. And again, the new cell comes, that also go through this and enter to the division. Again, the new cell come like this. This is called cell cycle. Now in cell cycle, what happens? The iterating cell cycle occurs in 5 stages. G0, in which the cell is not dividing but performs its normal work in the body. G1, or the gap 1, in which the cell grows. S, for synthesis, in which it duplicates its DNA. G2, or gap 2, in which it prepares to divide. M, or mitosis, in which the cell's nucleus and cytoplasm divide to form two daughter cells or two new cells. In human cells, they spend about 90% of their time in the G1S and G2 stages, which are collectively known as interphase. So 90% time interphase, 10% they spend on dividing. Most human cells complete the cycle in about 24 hours. What is 20? At the end of the day, you get all new cells in your body. This means each cell divided into two. Likewise, you are shedding off the old cells also, because your cells are dividing, doubling in 24 hours. So if you don't shed off those things, you will become double following day, right? Your size. So you can see all the cells are not dividing in 24 hours. That is one thing. But to fast growing cells, the cell cycle is much more speed. What you have to understand is there are a lot of old cells and you shed off all those cells, right? If you rub your hand and you get this something, there's some black color thing, maybe it is kind of holes are coming, rolling, right? What you think is this is kunu ambaranawa kiyala atulanawa. මේ kunu නෙමෙයි. These are cells. These are cells you are shedding off. Black color comes because of the dust you have maybe, but these rolls are not kunu. That will become a problem. No? So these are your cell layers. When you rub hard, when you are bathing, you are removing your top cell layer, right? Okay, most human cells complete the cell cycle about 24 hours. Fast-growing cells like those in the lining of the intestine may complete because we eat all the time. So with the food, cells are going away or being shed off. May come, so they are coming again and again. They are fast-growing, 9 or 10 hours. Liver cells take more than 1 year to grow. Neuron cells take many years to complete cell cycle. That is why you must have heard that nerve cells, if the nerve cells are damaged, it takes years to recover. Years to recover. So that is why, because nerve cells, cell cycle is very, very long, several years. Once a cell is prepared, made, that will retain for a long period of time, right? So this is the cell cycle. Next topic is cancer and the cell cycle. I will do it in the next lecture. Are there any problems? Can you, are there any questions? I hope you could follow the lecture. If there are unclear areas, Rohanti madam will explain you later. Okay. So I'm going to, what is this? No questions. Okay. Thank you very much then, and good night to everybody.
Recording stopped.